All right, moving to active transport. Active transport is going to require the use of energy. This energy typically is going to come in the format of an ATP molecule. One of the most common forms of active transport is a carrier-mediated active transport, which basically just means that the solute or molecule that's moving across the membrane is going to bind to a carrier protein. That carrier protein will change shape based upon the activation of an ATP molecule, allowing for the solute molecule, whatever bound, to move across the membrane. Most of the time, these carrier-mediated membranes or proteins are going to move these molecules against the gradient, against the concentration. So they're going to tend to move from low to high. Again, not always, but most of the time. One of the most common examples of a carrier-mediated active transport system is the sodium-potassium pump. The sodium-potassium pump is seen in the nervous system as well as the cardiovascular system and in other places. We're also going to see this format of transport with the calcium pumps. Calcium pumps are extraordinarily common with the skeletal muscle system as well as the cardiovascular system. And this is also going to be how amino acids, many of them, not all, but many, move in and out of the cell. So let's talk about the sodium-potassium pump. Here's just a handy snippet of both the passive and active systems and how they compare against each other. All right, let's talk about the sodium-potassium pump. So in order for a sodium-potassium pump to function, you need to have 3, not 1, not 2, but 3 sodiums bind to the protein. These sodiums need to be inside of the cell. These sodiums are going to bind. We're going to have an ATP come along and split off its last phosphate group. That phosphate group is going to bind, but that release of the bond is going to release energy. That binding and releasing of energy is going to change the shape of the protein, allowing the 3 sodiums to exit the protein on the outside of the cell, and the protein is going to stay there. It will not change its shape back so that it can pick up 3 more sodiums until 2 potassiums bind to the carrier protein from the outside of the cell, at which point the phosphate group will release, changing the shape of the protein back to its original one, allowing for the 2 potassiums to move into the cell. Why do we have this sodium-potassium pump? We have the sodium-potassium pump because sodium and potassium are constantly and easily moving through the membrane based upon the action or the concentration. Why does this matter? The sodium and potassiums on each side of the membrane are maintaining an electrical potential called a membrane potential to that barrier, and a change in that membrane potential will trigger various actions within the cell. This comes into play when we're talking about things like electrical impulses in a neuron or the cardiovascular system. So we'll talk about those in more detail with those systems. All right, so a couple other things in regards to sodium-potassium pumps. Sodium and potassium are ions. Ion concentrations are going to move water into and out of the cell. So we're going to have sodium-potassium pumps helping to regulate cell volume. Heat production. In the thyroid specifically, increasing the number of sodium-potassium pumps is going to increase heat. Heat is a byproduct of the energy bond releasing. So we can generate more heat. We don't talk about it that much though in that case, in that usage, but that is a function of it. As I mentioned, maintenance of a membrane potential. We talk about that more specifically with the neurological systems, or components I should say, but this sodium-potassium pump and the membrane potential is across all of them. All right, then the second form of active transport is vesicular transport. Basically, large particles are surrounded by a membrane in a bubble-like vesicle that allows for that vesicle to merge with the plasma membrane via the use of energy, allowing things to enter or exit the cell. If it is moving out of the cell, we call that exocytosis, and if it's moving into the cell, we call it endocytosis. Phagocytosis is a form of endocytosis. This is when we engulf large particles that are outside the cell using what are called pseudopods, or large protrusions would be the best term for it, large protrusions of the cell membrane and the cytoplasm within the cell membrane to wrap around and engulf bacteria or debris. Penocytosis is when that membrane, just the membrane, loops around and encloses water. It's also known as a cell drinking. Penocytosis brings in water and whatever's dissolved in the water. And then we have receptor-mediated endocytosis. Receptor-mediated endocytosis is going to be when a specific molecule binds with a receptor on the outside of the cell. The binding of that receptor will cause the membrane to engulf whatever is around that receptor, drawing it into the cell. This would be an example of phagocytosis. Once whatever was engulfed is inside the cell, it will be in a pouch, and that pouch will typically fuse with a lysosome. That lysosome will release its contents into the phagosome, is what that ingested particles pouch is called. Those will bind, releasing the enzymes into the phagosome, digesting whatever in there, and then quite typically that phagosome will then refuse with the outside of the cell, releasing what it broke down outside of the cell and not inside. Here's our penocytosis, phagocytosis again. And this would be an example of exocytosis, something inside. We made a carbohydrate chain, we made a protein, whatever it may be. It's in its little transport vesicle from the Golgi apparatus. It merges with the plasma membrane and releases its contents outside.